Hello everyone, this is Banabithi Bose. I'm a PhD student in Computational Sciences in Marquette University, Milwaukee, Wisconsin. Here I will talk about our recent project CTDPathSim, a cell line tumor deconvoluted pathway-based similarity in the context of precision medicine in cancer. In cancer research and drug development, human tumor-derived cell lines are used as popular model for cancer patients to evaluate the biological functions of genes, drug efficacy, drug side effects, and drug metabolisms. Using these cell lines, the functional relationship between genes and drug response have been studied. Also, prediction of drug response based on genomic and chemical features have been explored. Knowing the drug response on the real patients, however, is a more important and challenging task. To tackle this challenge, some studies integrate data from primary tumors and cell lines to find association between them. These studies, however, do not integrate multi-omics datasets to their full extent. Also, several studies rely on a genomic-wide correlation-based approach between cell lines without considering the heterogeneous cell population in bulk tumors. To address these gaps, we developed a computational pipeline CTDPathSim, a pathway activity-based approach to compute similarity between primary tumor samples and the cell lines at genetic, genomic, and epigenetic levels. We utilized a deconvolution-based algorithm to get cell type-specific DNA methylation and expression of each tumor sample. CTDPathSim has four main computational steps. In order to capture the accurate methylation signal, in the first step we employed a deconvolution algorithm EDEC and inferred each sample's deconvoluted methylation profile with different cell types proportions. For this purpose, we prepared a reference methylation profiles of different immune cell types from peripheral blood data. In the second step, we used bulk tumor gene expression data and computed sample-specific deconvoluted gene expression profile for each sample. In the third step, we computed sample-specific and cell line-specific differentially expressed genes, that is DE genes, and differentially methylated genes, that is DM genes, among the same cancer type cohort. Then, using sample-specific DE genes, we computed enriched biological pathways. Similarly, using cell line specific DE genes, we computed enriched biological pathways. In step 4, we used DE genes that were enriched in union of pathways of each sample cell line pair and computed a Spearman rank correlation using samples deconvoluted expression profile. Similarly, we used DM genes that were enriched in union of pathways of each sample cell line pair and computed a Spearman rank correlation using samples deconvoluted methylation profile. We then combined these two correlation scores to get a unified similarity score for each sample cell line pair. We applied our method on TCGA breast cancer and ovarian cancer datasets and cancer cell lines from CCLE database. We integrated DNA methylation, gene expression, peripheral blood raw methylation probes, and cosmic mutation data in our pipeline. We computed deconvoluted methylation and expression profiles of breast cancer and ovarian cancer samples based on different immune cell types. In this figure, we showed the Z-normalized similarity scores computed by CTDPathSim for breast and ovarian cancer samples with CCLE cell lines. We performed several tests to assess the similarity scores of sample cell line pairs computed by CTDPathSim. We compared CTDPathSim with the four state-of-the-art methods, namely TSI, TC analysis, SRCCM, and cell liner. Our computed similarity scores were found to be significantly associated with the responses of known cancer drugs between cell lines and samples. In this figure, we showed the drug response concordance with sample cell line similarity scores of the drug vinorelbine between breast cancer samples and CCLE cell lines with the computed similarity scores by 5 different methods. We would expect As the similarity score increases, the drug response matching should increase. Here, the matching percentage of the response of the drug vinorelbine increases as the similarity score increases only in CTDPathSim, whereas we don't see this concordance trend in other methods. Now in panel B, we show the box plot of drug response match and mismatch in low similarity thresholds of all 5 methods. Panel C shows these box plots in high similarity threshold. For CTDPathSim, there were higher number of drug response mismatch between sample and cell lines in low thresholds, where there were higher number of matching incidents in higher similarities. We see the higher number of matching in SRC-CM and cell liner. However, we don't see any difference in panel B. Overall, we don't see this concordance of drug response in other methods. We ran many other evaluations for our tool. For further information, please read the preprint of this work and visit our GitHub page. I thank my advisor, Dr. CTDPathSim Bose, for his guidance in this work. I also thank my lab members and our funding sources. And thank you all for your attention.